study of how basically biological information flows into cells and into organisms. And people, well, there are bioinformatics is one thing, but then there are a number of approaches, of methods, of subfields, I would say, that are used, which are completely different. And people tend to equate bioinformatics with the Human Genome Project, because that's when probably it became kind of popular, started becoming kind of popular. But in reality, it dates back to 1965, especially to the work of one scientist, Margaret Dayhoffs. And she basically started collecting protein sequences, which at the time were quite few, and storing them on basically on punched cards. So the then equivalent of USB keys, very old-fashioned USB keys. And so she made basically available these first database of protein sequences to researchers which would want to use them. And since they were on punch card, they could be processed by computers of that time and then used to start doing hypotheses. So then over time, the pace of development of bioinformatics has been pretty slow, with a few algorithms like Needleman-Wunsch, Schmidt-Waterman, and BLAS, which we still use, and you might know, you might have used, that were mostly focused on analyzing, comparing, and understanding sequences. Okay, and then led by further developments with, as I said, the completion of the Human Genome Project at the beginning of this century, which then led to a number of additional, let's say, subfields in which bioinformatics moves, so it's like structural bioinformatics, but also gene expression and gene networks. And so, however, the core focus for a lot of time, as I said, has been dealing with sequences. Okay, and the idea was to first of all, to being able to systematically compare sequences, so to try to understand what was different between organisms, for instance, between genes, but then also to try to understand the function, so to pinpoint which parts of a given sequence, think about promoters, think about UTRs, could actually bear a specific function in cells, and try to understand what this function was. So sequence analysis and genome analysis in most recent times is a very big topic. There has been a lot of work about informatics. And one of the most actual, I would say, topics in sequence analysis is that of trying to figure out how genomes works, and trying to figure out especially how where we come from and where we are going, meaning that what is actually changing with evolution over time, going to ancient genomes to archaic ones, to very, very ancient, to try to figure out what is the function of genes and how the human presence in particular, but not only, has evolved on Earth over time. And the talk by Abdurahim will deal with this topic. Another big aspect which has taken a lot of importance in the last 20 years is that of gene expression and gene regulation. So the availability of the human genome has led to us being able to pinpoint genes, to understand how transcripts are produced, how they are translated, and so to wanting to understand what was changing in transcription and translation, and in general in gene expression under different conditions, and in particular with the onset and potentially progression of many diseases, including cancer. And one way of looking at this is to use networks. Okay, networks are, let's say, a representation of biological phenomena that is interaction. So a network is built by nodes and by edges, where a node is a biological entity, a gene, a protein, a transcript, while the edges represent a kind of interaction, which could be, if you think protein-protein interaction, then an edge would be the fact that the protein A interacts with protein B, simply. In terms, if we think of this in terms of regulation, you might think that an RNA binding protein then actually is binding to an mRNA or to an encoding RNA to regulate its expression or its translation. If we think about messenger RNAs, for instance. And the importance and the usefulness of this approach is that it allows you to model and to analyze really a huge number of interactions altogether. And here you got a richness that can't easily be represented without networks. So the fact that you can't basically try to compute and understand how multiple regulators in particular can concurrently work to affect translation and in general gene expression. And this can also allow you to draw circuits. So to find out specific mechanisms that affect, I don't know, like in this case, the response to radiation through the p53, and to try to understand how these networks, or these small subnetworks, I would say, can go over, it can go wrong, can be altered by disease, especially cancer, and how we what we can do probably to try and fix how these networks are actually working. Okay, and all of this underlies the fact that the level of regulation within the human genome and transcriptome is so high that indeed we need absolutely computational methods to try to figure out what's happening. Otherwise, we would be confined to a very high level and probably condition-specific understanding of gene regulation. And this topic will be taken on by Nikolina in her talk. Just concluding, just wanted to suggest, if you think you're interested in bioinformatics, try to learn a bit more, try to understand how a bioinformatician reasons and what it does. You've got a lot of resources out there, but in particular, singled out these two courses, which are on the Coursera platform, and on the left for beginners, and the one on the right for people with a bit more experience. But they are free courses. You can go at your own pace, and they are of very high quality. And so if one is interested, they can probably start from their journey through bioinformatics. And with that, I'll thank you for your attention. And if you have any questions, I'll be happy to take them.
So I have one, Erik.
Yeah.
The courses you mentioned, they are free?
Yeah, they are totally free. Yeah.
Okay. So maybe at one point we can send the link for the people if they want to.
Absolutely.
Okay. Somebody else wants to say something?
I would like to thank Erik for a nice introduction. It will be easier to the participant to follow my talk after this nice introduction.
Okay, maybe if nobody has a question now, maybe they will be later. So who wants to start first? Nikolina or? So maybe Nikolina, because it is with this introduction.
yeah, because of the topic of the introduction.
I don't mind, and the schedule of Abdurahim's talk was noted as a first, but I don't mind. I can also start, whatever you prefer.
Okay, so maybe you can start.
Okay. Okay, I guess now I'm sharing my screen.
Yeah.
Okay. In my presentation, I will show you how can bioinformatical tools, specifically gene networks and visualization tools, help us to reveal potential anti-tumor drug mechanism of action. It can be simply said that all biological functions are controlled by a complex network of molecular interactions, and in disease state, this network is disrupted, and drugs are administered to restore balanced regulation. However, application of a drug means additional factor in an already vast network, and therefore research of the drug effect usually results with the biological data which are not that easy to understand. various bioinformatical approaches and tools can be used to analyze those data and help us in understanding. That's what we did in our research in of halogenated pyroxene. We used help of bioinformatical tools to understand its effect. Halogenated pyroxene has been topic of research of our group for a long time. It is patented as a substance with a potential anti-tumor and anti-proliferative effect. Its tumor suppressive effect was observed on different tumor types in series of in vitro and in vivo studies, but its mechanism of action is still not completely clear. In one of the recent studies, halogenated pyroxene was identified as an apoptosis inducer in leukemia cells. And results of the study indicated us that it would be of a value to further dissect the mechanism of pro-apoptotic activity of this compound in leukemia cells. But from that study, we got insight into the cytotoxic effects of halogenated pyroxene in leukemia cells, and also into the effect on apoptosis-related gene expression in cells. But we didn't get any details about which would give us possible idea about the mechanism of action. That's why I collaborated with life science informaticians, and we tried to get more information from the gene set expression data. And that collaboration resulted with publication eventually. And we, in that novel study, used gene expression data set targeting 84 genes that are implicated in apoptosis to identify the genes that are regulated by 4 different concentrations of halogenated pyroxene in leukemia cell line, UT7, and non-tumor cells, peripheral blood mononuclear cells. As this gene set comprises mainly apoptosis-related genes, we focused on comparing the specific pro-apoptotic mechanism triggered in the UT7 cells and PMBC cells, and the corresponding signaling pathways to elucidate underlying mechanism of halogenated pyroxene actions. Raw data set was converted into the log to a fold change format to be easily analyzed in bioinformatical tools. The differentially regulated genes were firstly visualized in the form of heat map and bumble plot using R packages pHeatmap and ggplot. After that, gene mania was used to construct a protein-protein interaction network for functional enrichment analysis. Gene ontology, enrichment, and KEGG pathway analysis was also performed using our package cluster profiler and enriched Whizbox. The Cytoscape app ClueGo and with combination with Cluepedia was used to analyze and visualize specific cluster of genes to dissect differences between the cell lines into more details. To support our findings, we also used the databases Reactome and Panther for additional results. So by applying described methods based on the analysis of transcript expression levels of a panel of 84 apoptosis-related genes, differences in the response of tumor and non-tumor cells treated with halogenated pyroxene was used to gain insight into the affected pathways and differences in response to halogenated pyroxene of leukemia UT7 cells and non-tumor BBMC cells. And basically, the obtained results indicate functional differences in the regulated genes following differential treatment of halogenated pyroxene. And in the first part of the results, which I will present you, the power of visualization is demonstrated. And in the first step of our analysis, all 84 targeted genes were included. Graphical representation of their differential response between the 2 cell types among all treatments was obtained in the form of a heat map, which is shown here in the slide. A color scale indicates downregulation and upregulation from blue to red color. And expression changes that do not p-value threshold, which was set to 0.05, are shown in gray and eventually from further analysis excluded. Besides that, different annotations of the genes are noted on the left side of the heat map. These annotations show whether the gene is classified as pro-apoptotic or anti-apoptotic, and whether it is included in the extrinsic or intrinsic apoptosis-associated pathways or in both. And the most notable result in this heat map is the result from the cells treated with 0.2 milligrams per milliliter of halogenated pyroxene, and that result shows that 14 anti-apoptotic genes were differentially expressed between the 2 cell cultures. Considering that result, And some similar observation for the treatment of halogenic Barks in that certain concentration in some of our other studies on other cancer cell lines. This particular treatment is explored in the more detail in our subsequent analysis. Dumble plot was constructed to visualize significant fold changes in this particular treatment of 0.2 milligrams per milliliter of halogenated Barksin against relative negative control of the UT7 and PBMC cell lines, in purpose actually to see how much is the variation. And most of these 54 genes showed similar pattern. They were downregulated in leukemia and upregulated in the PBMCs, but 7 genes, which are listed here in the slide, seem to behave differently as they were significantly upregulated in both tumor and non-tumor cells. Besides this data in the Dumble plot, again, there is a gene annotation which is related to the extrinsic-intrinsic apoptotic pathways and whether the gene is pro-apoptotic or anti-apoptotic. After these results, we wanted to further analyze the function of the genes associated with the genes that were observed as differentially expressed, actually. In Gene Mania, we deeper assessed the function of the 7 genes which stood out in the Dumble plot. In Gene Mania protein-protein interaction network, those 7 genes are connected with the in inner cycle with the 20 predicted proteins in the outer cycles, and this analysis revealed extrinsic apoptotic signaling pathway is the most significantly enriched. After that, we, in the next step again, included all differentially expressed genes in both cell types, and they imparted it in analysis, in the gene ontology analysis, and sunburst plot construction network was constructed. From this, we can see that top 5 biological processes include regulation of endopeptides, peptides, cysteine type endopeptides activity, extrinsic apoptotic and generally apoptotic signaling pathway regulation. The top 5 cellular component terms associated with the genes are membrane raft, membrane microdomain, membrane region, mitochondrial outer membrane, and organelle outer membrane. And molecular function, which were noted, are cytokine receptor binding, peptidase regulator activity, cysteine type peptides activity, and the protease binding activity. Also, KEGG pathways analysis here shows the 20 related pathways for the same number of the genes analyzed in the previous analysis, and TNF signaling pathway, NF-kappa beta signaling pathway, and necroptosis pathway from this analysis are the most relevant for this study. The other are not that relevant for the effects that we explored in this study. The Klugo functional analysis, in that analysis, 14 anti-apoptotic genes and unaffected genes in normal cells were compared, and the results of that comparison against the KEGG pathway database showed that the regulation of selected anti-apoptotic gene and pro-apoptotic genes concomitantly affects several pathways which are directly associated with the promotion of programmed cell death following halogenated Barksin treatment. And also from this analysis, it is relieved that largest number of targeted genes are regulated, but halogenated Barksin treatment is engaged in regulation of apoptosis, of course, since all the genes are related to apoptosis, but via 3 specific pathways, NF-kappa beta signaling pathway, and after that toxoplasmosis, and as I mentioned, direct apoptosis. Besides that, from this graph obtained through Klugo, we can see that those genes are also involved in the various processes within the other cancers, which kind of support some previous finding from previous studies. Since I mentioned that we researched the effect of halogenated Barksin on different cancer types, it's also give us a clue that it makes sense to further research the effects of the halogenated Barksin on the other cancer types. For further assessment of specific functional mechanisms explaining the halogenated Barksin effects on gene regulation at transcriptional level, we used Reactome and Panther, and the data from these databases revealed pathways and biological processes for the same gene setting which was analyzed in the Klugo for 14 anti-apoptotic genes which were downregulated in leukemia and unaffected genes in the non-tumor cells, peripheral blood mononuclear cells, and common adaptation among most significant pathways in leukemia were again regulation of intrinsic and extrinsic apoptotic pathways, also regulation of other types of cell death such as necrosis or necroptosis, again NF-kappa beta activity mediation, regulation of endopeptidase activity, and so on. After presenting all the results, I want to sum them up and give a possible explanation of the main ones. First of all, we can say that concentration of 0.2 milligram per milliliter of halogenated Borg-SIN treatment is once again proved to be most efficient. 7 genes upregulation indicates in the both cells indicates us that at least part of the apoptotic triggers are common in the both cell types. Though the analysis of specific gene sets, anti-apoptotic ones deregulated in leukemia and unaffected ones in normal cells, with the aim to dissect differential response in 2 cell types, reveals that 14 anti-apoptotic genes which are downregulated in leukemia are associated with NF-kappa beta signaling pathway. Considering that and other gene ontology cellular component terms which are associated, and also relevant data from the literature, we've assumed that halogenated Borg-SIN initiates a process of tumor cell sensation to pro-apoptotic activation, probably via the back independent apoptosis induction, and mitochondrial depolarization by ROS generation and trail-like activation, which is then followed by inhibition of NF-kappa beta signaling pathway. With this summary and interpretation, we can conclude that halogenated Borg-SIN increases propensity to apoptosis in leukemia UT7 cells, but not non-tumor cells in vitro. Also, that concentration of 0.2 milligram per milliliter is emphasized once again as the most efficient, and that its selectivity can be halogenated Borg-SIN selectivity can be explained by trail-like activation and back independent mitochondrial depolarization by ROS generation, followed then by permanent inhibition of NF-kappa beta signaling pathway in tumor cells. Based on these valuable conclusions about the halogenated Borg-SIN effect, our study proves that gene networks can help us to deeper assess anti-tumor drug effect, and besides that, it proves that interdisciplinarity and collaborations are extremely important in nowadays research, because my team consisted mainly of molecular biologists couldn't have reached the presented results without help of our colleagues bioinformaticians. Hence, I would like to thank all of my colleagues, molecular researchers from my home institution, University of Sarajevo Institute for Genetic Engineering and Biotechnology, as well to my colleagues, life science informaticians from Norway University of Science and Technology, to let me be the part of this valuable experience and this study, which was eventually also successfully published. I left here the link for our paper, but I am here to answer any questions and just discuss further. And of course, I want to thank you all for your attention and for understanding with the problems of it, sharing my presentation.
So who wants to start with questions?
I have one, if I may.
Yeah.
Okay. So I was wondering whether you also thought of clustering your protein-protein interaction network to find groups of proteins which might be, let's say, relevant, for instance, with one which is altered than the others which are impacted.
You want to share that photo too? No? In that photo of gene interaction, it was in a gene mania, right?
Mm-hmm. Yeah.
Okay. This one.
Mm-hmm.
Yeah, here we analyzed these 7 genes which were particularly upregulated in both cell lines. They were also of specific interest, and we predicted 20 proteins and analyzed associated functions, and of course, most of them, of course, every of them is associated with apoptosis, since this is the panel of the genes related to apoptosis, and we wanted just to dissect the specific signaling pathways related to apoptosis. And it was important to us. The important finding is that this extrinsic apoptotic signaling pathway, which is more the most enriched. And based on that, we concluded that probably the both cell types actually start the process of apoptosis in a similar way, extrinsic apoptotic signaling pathway, but healthy cells are, let's say, better fighters, and they are not sensitized by halogenated pyroxene, which pushes cancer cells further in apoptosis, and the healthy cell lines not. Okay. I hope that it's what you asked.
Thank you. Yeah, thank you.
Paulina, you can.
Hi. So I just want to say thank you for the great presentation. Congratulations on being the first fearless presenters in our series of webinars. My question relates to the thing that I think I didn't get something, so I've missed some information. How long did you treat the cell with this inhibitor? And I wanted to know if you're thoughts maybe on the process.
Sorry, I can't hear you well. I don't know about the others, but the sound is.
You don't hear me.
Yeah,
I can hear.
Paulina was asking for how long you were treating the cells with that inhibitor, I think.
Yes, exactly, and whether you know any valuable tools to monitor the dynamics of apoptosis response, because it's also it has different phases. Yes, so you might see some expression of some genes that it's upregulated in the short term, but then upon longer treatment, you might observe more resistance mechanisms that could be activated in the cell. So do you maybe plan to follow on that, or I don't know whether you're friends with this?
Actually, in this study, we did not. The cytotoxic of the effect are published in the other papers, and cells were treated for 72 hours, and we only analyzed data after 72 hours. But we are conducting currently one study, which are you indicated, we want to follow the, let's say, into time intervals, the effect of the halogenated pyroxene. This is the result only after 72 hours. So what happens in between, we don't know. We know that after 72 hours, healthy cells find a way to fight, and actually are not that affected with halogenated pyroxene, but cancer cells go into apoptosis. We only know after 72 hours, but in this particular data set, there are no information about that's what happens before. But I understand what you want to ask, and we also ask ourselves that, and we plan to dissect it into the time intervals to realize when and which this each gene regulator or whatever is activated.
Great, thank you.
Maybe I have a question. Maybe I didn't understand something. I heard, but maybe not really. I mean, maybe it's not, you didn't say that. You said that it doesn't work in tumor cells. At one point, you said that the drug doesn't work in tumor cells, or I misunderstood.
No, actually, it works in tumor cells. It pushes tumor cells into apoptosis, but let's say it's beneficial to the healthy cells.
Okay.
Let's say beneficial. It affects less healthy cells than cancer cells.
Okay.
Maybe I tell something after this confusion with presentation.
Maybe I didn't heard correctly. Instead of it doesn't work, you probably say it works. And I have another question. This is a lot of work and a lot of different techniques. Do you think that this can be used to follow the effect of the drug in a patient?
Yeah, I absolutely do now, because through this collaboration and through this study, I eventually realized the importance of bioinformatical tools, concretely gene networks and visualization tools. I mean, it cannot be used by itself, but it's really supportive to every other aspect of the research, whether in vitro or in vivo.
Yeah. Okay. Okay, I guess nobody else wants to ask question now. So thank you very much, Nikolina. This is a lot of work, but probably in bioinformatics, this is always a lot of work.
Yeah, and this was a collaborative work, so okay. I cannot take hold.
Okay, so maybe we can follow with Abdurahim.
Thank you so much. I have shared my screen. I don't know if you see it.
Yeah.
Yeah.
Okay, so good afternoon, everyone. My name is Abdurahim Kalajdzic. I work here at the Institute for Genetic Engineering and Biotechnology at Laboratory for Bioinformatics and Biostatistics. And currently I'm a PhD student at Faculty of Science, University of Sarajevo. First of all, I would like to say I'm excited to be a part of this first Early Career Investigators webinar. And I have the privilege of presenting a topic that is a part of my PhD thesis titled Unlocking the Past, Bioinformatics Tools to Dig into Archaeological and Degraded Specimen Data. Bosnia and Herzegovina, a relatively small country nestled in the heart of Balkan Peninsula in southeastern Europe, boasts a history that belies its compact size. With its history marked by both triumph and tribulation, Bosnia stands as a testament to the enduring power of history. Previous studies on genetic structure in Bosnia and Herzegovina were mostly based on investigating intragroup heterogeneity and interpopulation differentiation of contemporary population. That's why we have a well-known genetic structure of those populations. Logically, next scientific step would be to analyze genetic continuum of Harana population through time frame, starting with the prehistoric period through medieval times and finally to the contemporary populations. And basically, goal of this study is to assess the dynamics of those populations in Bosnia and Herzegovina throughout historical periods, while also simulating various demographic scenarios using artificial neural networks as part of deep learning analysis. Focus on this PhD thesis will be prehistoric period, while data stored in archaeodNA and contemporary genetic database within framework of Institute for Genetic Engineering and Biotechnology will be used. On this slide here, you can see a main idea of our project, where we assess the genetic continuum of populations through prehistoric period, then medieval, and finally contemporary period. Of course, taking into account different demographic events that occurred during this time. Assessing this genetic continuity is very interesting journey. Through advancements in genomics, bioinformatics, and archaeology, we can actually trace the threads of our genetic heritage. This exploration offers us a unique window into the migration patterns, adaptation of the population, and possible expansion or bottleneck of these populations. It also illuminates how ancient genes persist in modern day populations. Primary scientific question will be actually, does the genetic variation of prehistoric human population in this region constitute a part of the genetic structure of medieval and contemporary Bosnian populations. We actually hope that bioinformatics will give us answer to that question. We have used two different kinds of molecular markers. First is HTS-NGS, that's high throughput sequencing as part of the NGS technology. First, we have performed bioinformatics analysis of these short HTS sequences. Then we will be assessing the intergroup heterogeneity and intergroup variability between these three analyzed populations from prehistoric, medieval, and contemporary period. And finally, we will be utilizing artificial neural network in predicting the genetic continuum of these populations. Secondly, we have used mitochondrial DNA, and after sequencing HAVES regions, we have analyzed mtDNA polymorphisms and predicted the corresponding haplogroups. As you all know, probably deep learning has a vast majority applications in various analysis of biology, especially population genetic data. Here you can see a very simplified architecture of artificial neural networks that we'll be utilizing in this study, where basically those networks are comprised of three layers. Input layers that receives initial data, where each neuron represents a single feature, a single data. Then the most important part is this hidden layer, basically more than one hidden layers that are consisted of many hidden neurons that are performing the transformation of that input data. Number of these hidden neurons and hidden layers is very crucial, and actually depends on how complex is the analysis that we are about to perform. And finally, the output layer that provides a predicted results. We have actually already used artificial neural network and deep learning methodology in 2021, where we published a paper where we validated forward in time method using artificial neural network. Probably those of you who are not familiar so much with population genetics, forward in time is actually a method that is used for simulating the evolution of the population. And we basically now introduced artificial neural network in validating those results. And actually results were very surprisingly good. And they validated the results obtained with the forward in time method. In regards to the deep learning of the processing of these HTS sequences, as you all know, HTS, high throughput sequencing, is basically sequencing multiple DNA molecules in parallel and generating a vast number of short DNA sequences. HTS sequences from prehistoric data will, in our study, be used as a pattern for medieval and contemporary data. As you all know, probably artificial neural network has different methods of applying to different data. One of them is actually feed forward that we'll be using in our study, where information travels in only one direction, from input neurons through the hidden layer to the output neurons. There are multiple advantages of using this type of artificial neural network, but in our case, main advantages are that the output information from the respective layer does not affect the same layer, and that's very important to us. And also in regard to the fact that the archaeological DNA is very degraded due to its being so old, one of the biggest advantages is that the feed-forward network is able to recognize certain patterns in these archaeological degraded samples. Here you can see basically a graph that shows how are we going to implement this artificial neural networks in obtaining different sets of results. We have comprised our results in 3 different sets. First, we'll be using prehistoric NGS data as training data. Then we will be taking into account different demographic events and until up to the medieval period. And then we'll obtain output for medieval NGS data where we will be actually comparing that scenario predicted data with actual medieval data that is obtained in the ArchaeoDNA database of Institute. Secondly, we will be using, again, prehistoric NGS data with different demographic events taken into account up until contemporary period. And after that, we will compare that scenario predicted contemporary data with actual contemporary data that is available here at the Institute for Genetic Engineering and Biotechnology in our database. And finally, we'll take medieval NGS data and compare that to the take into account, of course, still the demographic events up until contemporary period, and then compare that to the actual contemporary data. So those are the three main sets that we will be utilizing artificial neural networks. It's worth mentioning, of course, that before this project, we had one more project called Genetic Characteristics of the Population of Medieval Bosnia in 2019, where we only taken into account the medieval period and not NGS data, but autosomal and YSTR markers. And the results of that study showed that the absence of statistical significant genetic differentiation between the medieval and contemporary Bosnian population based on those two sets of markers can be observed. Main goal was actually to understand the population structure of those living in this area. However, archaeological research confirms the existence of Paleolithic populations, and considering that the mentioned area within the broader geographical region of Balkan Peninsula is very interesting from the migratory perspective, additional research would certainly complement information about the way of life, genetic structure of the prehistoric population. And therefore, information about the genetic diversity of the population of that time is still not available, and explaining these details would certainly contribute to the new insight about the genetic continuity of population in Bosnia. We have actually divided our project into 4 phases. First and second phases are already performed. Those include sampling of archaeological human skeletal remains, and the second is laboratory activities on isolation, amplification, and genotyping using different sets of molecular markers. The third and fourth phases are currently undergoing, where we'll be comparing population genomic comparison between these 3 populations, and finally applying the artificial neural network in understanding that genetic continuity across different historical periods. As I already stated, first phase sampling of archaeological human remains was already performed. You can see on the left side here a 700-year-old skeletal remain from the medieval period that was already performed during the last 10 years. And ArchaeoDNA database of Institute has DNA profiles of 150 individuals from this medieval period. However, for prehistoric period, sampling of archaeological skeletal remains was performed in 3 sites, Kopilo, Gradišće, and Gradina Pročica. Those are small villages near Zenica town in middle Bosnia. And all of this, of course, work was done in cooperation with the Faculty of Philosophy Department of Archaeology. Second phase included the laboratory procedures in the context of preparatory phases like sterilization, cleaning and washing samples, extraction and quantification of DNA, generating NGS data, and sequencing and analysis of mitochondrial DNA. Third and fourth phases are currently undergoing. In regards to the mitochondrial DNA analysis of medieval period, we have currently 1 paper under review where we basically sequenced Haves 1 region from 32 samples. And interestingly, all 32 samples had H haplogroup, 30 of them had H2A, and 2 had H5. Of course, we have provided and performed extraction and all the analysis twice just to be sure. And also we provided autosomal DNA profiles that show that there is no cross-contamination for these samples. In the context of bioinformatics tools, we use BioEdit for aligning, cutting, and trimming sequences, and haplograph BA with fill of 317 for haplogroup prediction. Of course, Metcalfe and PAS were used for statistical analysis. And we obtained results that showed that the medieval Bosnian population in context to the contemporary populations of Bosnia had a difference between 46.7 to 53%. And in the context of other medieval populations, we obtained results that showed that the smallest was with the Italian medieval population. As you can see here on the picture, this is the neighbor joining dendrogram, and it actually shows where the medieval Bosnian population is positioned in the context of other medieval European populations. Of course, this does not mean that we have much similarities with Italian and Spaniard medieval populations, but to emphasize the fact that the medieval Bosnian populations was a very close population, and that the genetic influx of other populations was not so emphasized. In regards to the mitochondrial DNA analysis from prehistoric period, that process is currently undergoing. In regards to NGS analysis, we have performed NGS analysis for medieval data. We performed quality control using FastQC and FastQE, and mapping with HISAT-BA. All of those softwares are actually available through Galaxy web servers. And as you can see here from these pictures of FastQC, we have obtained very good results, taking into account that the DNA in question is 700 years old. In regards to the NGS analysis of prehistoric period, that also is currently undergoing. And finally, population genomics and comparison. We'll be utilizing different kind of softwares in that context. In the context of population genomics comparison, we will be using NGS PopGen, that is again freely available software to that is being used for measuring heterogeneity of certain population. Then ANGSD for allele frequencies, and NGS admix. Those of you familiar with population genetics certainly know the admixture model that is being used by, for example, structure software, and that software is basically positioning and clustering the population in different clusters based on their genetic variation. And finally, we will be implementing artificial neural network in analyzing these genetic continuity. We will be using our programming language and specific packages that are created for artificial neural networks, and those are Labra and Neural Net. And finally, I would like to thank our team here at the University of Sarajevo. This is a very multidisciplinary project that includes a vast majority of faculty, such as Faculty of Philosophy, Department of Archaeology, Faculty of Dentistry with Dental Clinical Center, Faculty of Electrical Engineering, and of course my home institute, Institute for Genetic Engineering and Biotechnology, Laboratory for Bioinformatics and Biostatistics, for Forensic Genetics, and for Human Genetics. I would like to thank you for your attention. If you have any questions, please feel free to ask me. And of course, I know that we are like on a time schedule here, so I provided a email if you have any further questions. So yeah, thank you again.
So time for question is open.
I can ask a quick question.
Yes, sure.
Very nice, Abdurahim.
Thank you.
We're having problems with your name, but I hope after we try to repeat it some 20 times, we're getting there. Yeah.
Yeah.
Have you been involved in the extraction of DNA from the samples? I mean, these
Yes.
archaeological samples in particular.
Yes.
Okay, cool. Another way to put it. How do you avoid contamination? You know, cross-contamination with environmental DNA.
Well, the fact that the laboratory for forensic genetics and basically all archaeological DNA samples require specific laboratory procedures and specific laboratory space. So basically, here at Institute for Genetic Engineering, we have a laboratory just for extracting archaeological DNA samples. Of course, all of those area are cleaned with alcohol, different chemicals, just to avoid the cross-contamination. Of course, we always perform the analysis twice just to be sure, and we also have autosomal DNA profiles of all the persons that are working in that laboratory, just to confirm that the DNA in question is not contaminated.
Okay. You have a profile for all your colleagues too. That's
yes.
Cool.
Yes.
All right. How the fragments of DNA you extract, are they really, really tiny? I mean, these are very silly questions in a way, but I have no idea about any of this. So,
are you talking about the archaeological samples or DNA samples?
the DNA samples. Yeah.
Oh, the DNA. Well, yeah. Well, it's safe to assume like that the skeletal remains that are in question are 700 to 3,000 years old. So the DNA will be extremely degraded. It will be in very short fragments, but very surprisingly, those results are still possible to perform analysis on them. And of course, with these medieval period, we have published several papers in that regards. And yeah, so even though that samples are very degraded, it's still very usable.
All right, thanks a lot.
No problem. Thank you. Any more questions?
Yeah, I have a question.
Yes.
Does these studies could show, for instance, a pathology in the population linked to their history or something like that?
Well, it all depends on the quality of DNA extracted from these samples. But of course, our project will also take into consideration some of the possible pathologies that the population in that time had incurred.
And you don't have any study on methylation or epigenetic or something like that?
No, currently no. Just this NGS data and mitochondrial DNA with the analysis, with the utilizing artificial neural network in assessing the genetic continuity of the population here in this area. Just to confirm whether the contemporary population is similar to the medieval population and the prehistoric population here in this area.
Yeah.
I may add to the question about the pathologies, since this is the interdisciplinary project, we have all the skeletons evaluated by forensic anthropologists, and they already, for some medieval period skeletons, we have already an estimates of certain immunological disorders that we are going to tackle in future should Abdurahim finds out the way how to extract the data. Of course, you know that the highly fragmented DNA from archaeological specimens cannot be used for long reads, for long fragment reads, but more for SNP-based or short fragments analysis. So after Abdurahim validates these data, then maybe we can try to extract some medically relevant genetic information.
So
yeah, sorry, I interrupted you, Abdurahim. Maybe you had to add something more about this.
No, you emphasized it perfectly. The main goal will of course be the analyzing the genetic continuity. However, after that, we'll certainly take into account the possible pathologies that those people in that time occurred.
Erik had some question, or you were raising your hand.
Yeah. Just as a curiosity, do you have a way, or could there be a way of without? So, for instance, in your lab, you said you have the genomic profile for your colleagues, so you can exclude reads from them. But from general point of view, is there a way of identifying, of understanding whether a given read is from a more ancient genome rather than a more recent one? Is there a tool that can predict that?
I'm sorry, I didn't hear the last part of the question.
I'm sorry. Yeah, in general, I was wondering, is there a tool or an algorithm that can predict whether a given read, given sequencing read, comes from is more likely to come from a recent genome, so modern time genome, rather than an older one?
Well, I don't think that there's any algorithm for that specific question, but I do believe that the contemporary DNA would certainly be of much more quality than the archaeological DNA. So in the start, you will definitely know the difference in that context. But to answer that question, I really do not think so that there's any algorithm or software that actually extravates the archaeological DNA and the contemporary.
Okay, thank you.
No problem.
And maybe just to clarify, so the most of these specimens are taken from the graveyards, which are distinguished by the way of burying bodies that were in sort of the stone caskets and closed. So I mean, they are archaeologically completely evaluated. So practically there was no risk for cross-contamination during the history, but more at the point of excavation. So and we try to avoid that because the people from forensic laboratory genetics participated the process from the uncovering the bodies to the lab. Thank you. And yes, I would like to thank Sophie on my behalf and on behalf of the institution for an invitation and an opportunity to present the results of two of our young colleagues, PhD students. We have more of them in the various areas of molecular biology applications. But since you were interested in bioinformatics application and bioinformatics tools, we selected these two our great exceptional students. So thank you very much, Sophie, for this opportunity. I think it's a great idea for young people to get to know each other. At the end, we are all close.